Okay, we'll go on to look at what is the genetic code. Okay, so our genetic information is stored in the form of a nucleotide sequence, right? ATCG in the form of DNA, and then it's transcribed into the nucleotide sequence of the mRNA, right? AUCG. So in translation, right, this genetic information is going to be used to make a amino acid sequence, which is no longer ATCG or AUCG, it's amino acid sequence, not nucleotide sequence. So how do we read the nucleotide sequence in order to know what is the amino acid sequence in the protein, right? How do we translate the nucleotide sequence to the amino acid sequence? Right, so the rules for translation is known as the genetic code. Okay, so we call the central dogma how genetic information flows from DNA to mRNA to the amino acid sequence of the protein. The mRNA, right, is going to be read by the ribosomes in the form of codons, which are a triplet code, which is so each codon is consisting of 3 nucleotide sequences. Right, so the mRNA is read in codons in the 5' to 3' direction. We'll learn this later in translation. That's the next lesson. Okay. So each codon, right, is going to specify an amino acid. This is how we translate the nucleotide sequence in trees into the amino acid sequence. So each of the 3 nucleotide sequences as a codon would code for a particular amino acid during translation. So if I see the nucleotide sequence is UUU, I know then therefore the amino acids must be phenylalanine, for example, or GGC must be glycine. Right, so this is like the how to read the mRNA. All right, so this set of rules is known as the genetic code. So let's look at some features of the genetic code. Firstly, as mentioned, right, it's a triplet code. So the mRNA is read in triplets, right, 3 nucleotide sequences as a codon. Okay, reason is because there are 4 types of nucleotides, right, in the mRNA, AUCG, and there are 20 amino acids, right? So if we want to use the mRNA sequence to code for the amino acid sequence, if each codon, or if we read the mRNA sequence one by one, right, then each of AUCG, if they each codes for a different amino acid, they can only code for 4 different amino acids. Or if we read the mRNA sequence in twos, right, so let's say AU codes for one particular amino acid, AC codes for another particular amino acid, and then UC another particular amino acid, maximum is only 16 amino acids that can be coded for, because there are only 16 different combinations of codons if each codon only consists of 2 nucleotides. Right? But if each codon consists of 3 nucleotides, then that would allow for 64 different combinations of codons, and that would be enough to code for 20 amino acids. Okay, so that's why the mRNA sequence is read as codons, each with 3 nucleotides. Okay. I have an animation to explain that if you did not understand verbally. All right. Also 16 different codons if each codon has 2, right? But 64, as you can see in the codon table, right? If each codon is of 3 nucleotides, so therefore a triplet code. Okay. So if you look at all the 64 codons, right, not all of them codes for amino acids, right? There are 3 UAA, UAG, and UGA that are called stop codons. That's where it signifies the stop, the termination of translation. You look at it next lesson when you look at the process of translation. Okay, but for now, right, you need to remember the sequence of the stop codons. Right? So UAA, UAG, UGA. There's also a start codon, right? AUG here. Okay, if you can see my cursor. AUG, which also codes for a methylene amino acid. Okay, so start codon, but also codes for amino acid. Stop codons don't code for amino acids. Okay, so that means that out of 64 codons, only 61 codes for the 20 amino acids. Okay. The next feature is that the genetic code is universal, right? So almost all organisms on Earth, at least, right? Almost all organisms follow the same rules. Okay, so if CAG codes for glutamine in human cells, it will also code for glutamine in all sorts of other cells, right? Or another example, say here, if UUU codes for phenylalanine, then if I put the same UUU into a bacteria, it would code for phenylalanine. I put the same UUU into, say, a plant or an animal or whatever other cells, yeah, then it will still code for UUU. Right? So almost all organisms follow the same code, with the exception. Okay, so scientists have found some yeast which deviate a little from the genetic code. Right, so the codon CUG, instead of leucine, it codes for another amino acid in some other yeast species. Right, so this is like an exception, right? But even the exception is only exception in one codon. Right? So all the other codons in these species of yeast would also code for the same amino acids as all the other organisms. Right? So it's just exception in one codon, so far identified. Okay, so going back to the features of the genetic code, right? The genetic code is described as degenerate, but not ambiguous. Right, so being degenerate means that more than one codons can code for the same amino acid. Right, so remember, right, there are 61 codons that codes for amino acids, but only 20 amino acids, right? So that tells you, right, some codons surely codes for the same amino acids. So if you can see, right, proline here, I'll just take this example, proline, right, 4 different codons codes for the same amino acid, proline, and they differ only at the third base. So it always starts with CC, right, but then whether it's U, C, A, or G, right, it still codes for proline. Right, of course, there's exception, right, leucine is one. Okay, so you have this one, these 6, right, codons all codes for leucine, but they don't only differ in the third base, right? Especially for this UU, which is different from the CU. There's another exception would be the serine, right? 4 here and 2 here. Right? So I think these are the 2 exceptions lah. Other than that, right, all the other amino acids, if they have multiple codons, the codons only differ in the third base. Okay, so that's the degeneracy, right? More than one codons code for the same amino acids. But it is not ambiguous, meaning if you look at each codon will only code for one amino acid. Okay, I hope this is not too confusing. Right, so if I look at, say, CUU, right, CUU can only code for leucine. It doesn't code for something else, right? But if I look at leucine, it has more than one codon. It has 6 codons that codes for the same leucine. Okay, so that's degenerate but not ambiguous. Okay, and lastly, the feature is that it is non-overlapping, meaning if I read the sequence of mRNA in trees, right, starting with the start codon AUG, which codes for methylene, I cannot read the next codon as UGG because this overlaps. Right? I also cannot read GGG, which overlaps at the G. The next codon has to be GGA, which codes for glycine. The next one has to be another AUG, which would be another methylene. It can't start again, right? But you just codes for another methylene. And then UGU, which codes for cysteine, and then UAU, and lastly the UAA, which is a stop. So no amino acid here. So this is actually how translation occurs, right? So if you can see, I'm combining all the features together, right? I have the triplet code. Each triplet code only codes for one amino acid, so that's not ambiguous. Right, I am not showing the degeneracy here, but I also showed the start and stop codon. So this is actually how the translation process will occur, which you will learn again in the next lesson. Right, so but anyway, this is to show you that the genetic code is non-overlapping. Right, so when you read the next codon, you cannot overlap with the previous one, and of course there's no gap or so you can't skip a nucleotide. Right, so it just follows. Right, so that would be the genetic code normally, but then scientists like to make new things. So there are scientists, I think in Japan, that has made a synthetic DNA with 8 different bases. So they gave it a Japanese name, Hachimoji. It sounds quite nice, which means 8 letters. Right, so this DNA is fully functional. It can be transcribed into mRNA. Okay, but it's in the lab, of course, because these are synthetic DNA. So our cells, the normal enzymes in the cells, don't recognize these new DNA lah. So it's but in the lab it can be transcribed into RNA that also have 8 different bases. Okay, so think about this, right? If DNA has 8 different bases, but each codon, right, only have still have 3 nucleotides, right? So it's the same triplet code, but now we have 8 different types of nucleotides, right, in the RNA. How many codons would there be? Right, so there will be 512 different codons possible, right? Because the formula is x to the power of y, right? X meaning x would be the number of different nucleotides, and y, the power, would be the number of nucleotides per codon. Right, so in this synthetic RNA, right, there are 8 different nucleotides. We still assume that each codon has 3 nucleotides, so that would make 512 different combinations of codons, which would obviously be too many lah. Right, so that would be the end of the genetic code.